In this part, we are going to discuss about the internal structure and functions and activities that is taking place within the seminiferous tubule. And also, before knowing about the seminiferous tubule and its internal structure, we shall recall what we have discussed and studied in the previous class. In the previous class, we had discussed about male reproductive system. The male reproductive system consists or comprises of primary as well as the secondary sex organs. The primary organs or the primary male sex organs is nothing but a pair of testes, and the secondary organs are the organs which are helping or accessoring the production of sperms within the testis, and those accessory organs or the secondary reproductive organs is nothing but the ducts as well as the different glands and the external genitalia, that's nothing but penis. And we shall recall the structure because that helps us to study the next concept. As we said, the primary sex organs, they are nothing but a paired testis. Two testes are present, and the testis is completely covered by the layer that is called as tunica albuginea, and these layers are forming a thick coat or covering around the testis, and the testis layers are entering inside or internal region or surface of the testis, and due to the extension of these layer inside the testis, they are forming a cross walls or like a septa. They are forming the septa or the cross walls like structure. Due to the formation of these cross walls, due to the extension of the membrane, the formation of septa took place, and due to the septa formation, these septas are forming many compartments within the testis, and those compartments may be approximately 280. So what we can say, a single testis consists of 250 compartments, and those 250 compartments is nothing but the testicular lobules. The compartments are called as testicular lobules. These testicular lobules, I am enlarging here. Within the testicular lobules, there is a presence of highly coiled 1 to 3 in number. They are nothing but seminiferous tubules. 1 to 3 highly coiled tubular structure is present within the testicular lobules, and that structure is called as nothing but the seminiferous tubules, and these are the actual region where the spermatogenesis takes place. And how the seminiferous tubules is? The seminiferous tubules are highly coiled in nature, and as soon as the seminiferous tubule are continued and reaches the end, they are becoming straight. In the initial region or in the beginning part, these seminiferous tubules are highly coiled, but gradually the coiled nature is not observed. Instead, the tubules are becoming straight at their end, and that straight ends of the seminiferous tubules are connected to a next duct or the next tubule, which we are calling it as rete testis. The seminiferous tubules ends and it releases the sperms to the next duct, that is rete testis, and rete testis is continued by the next duct which receives and collects the sperms, that is nothing but vasa efferentia. These vasa efferentia are further continued and they opens at the epididymis. All the vasa efferentia, all number of vasa efferentia are connected to the epididymis, and the epididymis is next further continued to form vasa deferens. Vasa deferens is also called as urinogenital tract. Why we are using that term? Because the same vasa deferens is carrying both the urine, because it is connected to the next ureter, as well as it is carrying the sperms during the time of reproduction. Urinogenital tract, we use the term, and that is connected and to the seminal vesicle. It is continued by the next glands, and that seminal vesicle is connected to the ureter, that is nothing but the urethra, which carries urine during the time of excretion of urine and which carries sperms during the time of reproduction process. This is how the actual connection is, what we discussed in the previous class. But now what we are doing, or what we are studying today, is the testis is sectioned and we are observing the testicular lobules, and within the testicular lobules we are entering inside the seminiferous tubules. To study a clear picture of seminiferous tubule, we have sectioned the seminiferous tubule here. So transverse section is done of the seminiferous tubule. When we observe the sectional diagrammatic view of seminiferous tubule, the seminiferous tubules, the word itself is telling it's a tubule. That means it is a tube in nature. And when we observe the seminiferous tubule in a section, they are scattered. 2 to 3 seminiferous tubules are scattered in a single lobule. So we have taken the section here. A seminiferous tubule is composed of, or it is made up of, germinal epithelium. It is made up of germinal epithelium cells. And when we take the sectional view, the outermost lining is composed or made up of germinal epithelium, and the germinal epithelium cells is differentiated into 2 different cells. Which are those 2 different cells? One is spermatogonia and other one is Sertoli cells. The germinal epithelium is forming the outermost lining of the seminiferous tubule, and the germinal epithelium is of 2 different types of cells. 2 different types of germinal epithelium cells is found in the seminiferous tubules. On which basis we are differentiating into 2 different types? That is, the first type, as we said, spermatogonia, is one of the male germ cell, and the second one is Sertoli cells. These are the 2 different germinal epithelium cells that we found in the lining. How they are differentiated? Because these germ cells, male germ cells, are the spermatogonia cells. These are the cells which gives rise to the sperms, or which give birth to the sperms, and those cells, that is the spermatogonia cells, will undergo meiotic cell division. Why we are using the term meiotic division? Because these diploid cells will undergo meiotic division, that is reductional cell division will be seen, because the gametes are always in the haploid condition, and that haploid cells which are arising from the spermatogonia has to be formed by the meiotic cell division. The number of chromosomes will be reduced, and these spermatogonial cells will undergo meiotic cell division to form the next stage of cells, that is nothing but primary spermatocytes. They undergo meiotic cell division, and further next development of the cells that can be seen, which is formed by spermatogonia, is next comes the primary spermatocyte. And this primary spermatocyte, again it will undergo development to form spermatids. Always the spermatids are in haploid condition. We'll discuss in detail when we study about the spermatogenesis. What we must know here is the germinal epithelium forming two different types of cells. One is male germ cell or spermatogonia, and the second type of cell is Sertoli cells. The spermatogonia will undergo differentiation and to form primary spermatocytes. The primary spermatocytes will further develop into, or it will form into, spermatids, and these spermatid cells will develop into sperms. They will develop into sperms. To cross these stages, these cells or the spermatids, to convert into a developed sperm, it needs nourishment, and that nourishment is given by the Sertoli cells. The Sertoli cells, which are present in the germinal epithelium, they are nothing but the modified cells. Sertoli cells are also the epithelium cells, germinal epithelium cells, but they are modified. For what they have modified? For the purpose of nourishing the developing sperms. They are modified, and these Sertoli cells will provide nourishment and nutrient to develop the spermatids into a mature spermatozoa. And because of providing nourishment, the Sertoli cells are also called as, the Sertoli cells are also called as nurse cells. They are also called as nurse cells. That is, the Sertoli cells will provide nourishment to the developing sperms, and along with the seminiferous tubules, there are other remaining seminiferous tubules, and these seminiferous tubules are fixed or connected with the tissue called as connective tissue. It is called as connective tissue, or the connective tissue is also called by another name, that is interstitial tissue. They are present in between two different seminiferous tubules. They are connecting, they are giving bonding or connection between the different seminiferous tubules, and that tissue which is present between the seminiferous tubules, which is present in the external surface and which is present between different seminiferous tubules, these tissues are called as connective tissue or interstitial tissue. And in the interstitial tissue, there is a presence of some deposition of cells at some intervals of space between the different seminiferous tubules, and those cells are called as Leydig cells. The cells which are present between two different seminiferous tubules in the connective tissue, and those cells are called as Leydig cells. The Leydig cells are also called by another name, that is interstitial cells. The Leydig cells are also called by another name, that is interstitial cells. Why we are using the word interstitial cells? Because they are present in the interstitial tissue. They are present between two seminiferous tubules. If so, what is the function of interstitial cells or the Leydig cells? The main function of the interstitial cells or the Leydig cells is to produce male hormones, male sex hormones, male sex hormones such as androgen or testosterone. Male sex hormone or androgen or testosterone, they are the alternate names for the hormones, and that androgen or testosterone is produced and synthesized by the Leydig cells, and these are the hormones which controls and regulate the formation and development of the spermatogenesis. So this completes about the sectional view of seminiferous tubules. We shall brush up what we have discussed today. We studied about the seminiferous tubules, sectional diagrammatic view of seminiferous tubule. A seminiferous tubule is scattered in the connective tissue. The number of seminiferous tubule is for around 2 to 3 in number in a single compartment or in a single testicular lobule. And when we take approximate number of seminiferous tubule, there may be 900 of seminiferous tubules or 750 to 900 of seminiferous tubules in the single testis or in a one testis. Here we have taken the section and they are scattered. The seminiferous tubules are scattered in the connective tissue. It is held and it is giving connection and fixed with each other. The seminiferous tubules are connected by the connective tissue or the interstitial tissue. The seminiferous tubules are spherical or oval in shape, and the seminiferous tubules consist of germinal epithelium cells as the outermost lining. As the outermost lining, germinal epithelium cells is present, and those germinal epithelium cells is giving cover or the outer coat lining for the seminiferous tubules. And there are these germinal epithelium cells are differentiated into, one is spermatogonia and other one is Sertoli cells. Spermatogonia cells will further undergo meiotic division to form the sperms, and the spermatogonia is develops into primary spermatocyte, and the primary spermatocyte will undergo meiotic division and forms the secondary spermatocyte, and that secondary spermatocyte further develops into spermatids, and these spermatids will develop various structures and becomes a mature spermatozoa, and these mature spermatozoa are developed to become sperms, that is the spermatids are provided with the nourishment, and that nourishment is given by the Sertoli cells. They are providing nourishment for the developing spermatids and the developing sperms, and they provide nourishment and transforms the spermatids into a mature spermatozoa. And in the connective tissue we can find mass of cells. There are, at some intervals, there is some deposition of cells, and those cells are called as Leydig cells. Leydig cells can also be called by another term, that is nothing but interstitial cells. These interstitial cells play a very important vital role in the production of male hormones or androgen or testosterone. These are the hormones which are very essential and which controls and regulates the production as well as formation and the development of the sperms. And this hormones completely regulates the function of the seminiferous tubules. And we have completed the concept and the sectional view of seminiferous tubules.